This is Tulsi from BioBeats. All test cross are backcross, but a backcross is not a test cross. What do you mean by this statement? What is test cross and backcross? Where do we use these two crosses in our day-to-day life or by plant breeders? So today let us discuss about test cross and backcross in genetics. So when we talk about a tall plant which is dominant, so the phenotype of this plant is dominant. That plant genotype can be of any one of these, either capital T or capital T small t, right? So we cannot give guarantee like when we observe a tall plant, it must be a pure, may not be. It can be a heterozygous, right? So what makes the different if it is with the homozygous dominant or heterozygous dominant? Because in the next generation, when it is crossed with the pure dwarf, the percentages of the offsprings with both the parental characters will differ. So let us check that with one case. So see, this is the pea plant. One is tall and the other plant is dwarf. So when we check the tall parent, it might be having either capital T or capital T small t. So that means when we observe a tall plant, that will not give you clarity about its genotype. It gives only clarity about its phenotype. But when we observe a dwarf plant, it gives clarity about both phenotype, genotype, because this dwarfness is recessive feature. When is it expressed as a phenotype? When it has both recessive alleles, right? Why? The reason behind this is if both the alleles are recessive, then they can express their feature. Otherwise, if they have one dominant allele, that suppresses this recessive allele. So that is the condition, right, which is given by Mendel. So when we check in this case one, we have seen the tall plant, but its genotype is capital T small t. And you check the case 2, where this tall plant has the genotype capital T capital T. That means here this tall plant is heterozygous. It's not pure tall. And here it is pure tall, right? So that's the reason when it is having capital T small t and the dwarf plant small t, observe the offsprings, capital T small t tall, capital T small t tall, and small t dwarf, small t dwarf. So that means 50% we got tall and 50% dwarf. Then coming to the case 2. Here we have taken a tall plant, but which has two pure alleles, right? So capital T, capital T. So in this case, we have capital T small t tall, capital small tall, capital small tall, capital small tall. So that means all these offsprings are tall, right? Even though you have taken phenotype same, but you got the offsprings different, right? So how do you know this procedure? This is by backcross and test cross. So backcross, its name indicates we are taking one unknown genotype that is F1 hybrid. How do we produce F1 hybrid? Again, go back to monohybrid cross, where we take a pure tall plant, cross with pure dwarf plant. Whatever we get in F1, all are hybrids because each one will have capital T, small t, right? So we have taken that hybrid. That means we are taking a offspring and we are crossing with one of its parent. That's why we do call it as backcross. So in case of backcross, we do cross with one of these parents, which is definitely it is a dominant parent. So that's the reason you can check here the gametes with the alleles capital T small t, capital T, capital T. Right? So now here when you check the offsprings, it receives capital small, capital small. So what is the phenotype of all these four offsprings? That means four possibilities. All these are tall. So that's why resulting ratio is all are tall. Clear? So how is it useful? Generally, plant breeders, if they want to get all their offsprings in the coming generations with the dominant feature, they can take up the F1 hybrid and cross with one of its parent who is purely dominant, right? Then, what do you mean by test cross? Its name indicates we are testing something. What do we test here? We are going to identify the unknown genotype of that parent. How do you know? Here, we cross the F1 hybrid with homozygous recessive plant, right? So when we cross with heterozygous with homozygous recessive, small t, you can check here, capital T, small t, tall, small t, dwarf, capital T, tall, and small t, dwarf. That means what? Ratio is 1:1. That means 50-50, tall and dwarf. So from this, we do identify when we get this feature, it shows that that unknown genotype of this F1 is heterozygous, right? So test cross is homozygous recessive individual. We are crossing with the genotype of the unknown individual. So which will be relieving their or relieved by their offspring. So let us take one example. For example, we have considered here again garden pea, which has the dominant feature purple color flower and recessive feature white color flower. Generally, if it is purple color flower, it must have at least one dominant allele. Otherwise, both can be dominant. So you can see here, it can be capital P, or one capital and one small. But coming to white color, both should be recessive, that is small p, right? Now, we want to know the first one, which is dominant, whether it's a purely dominant or otherwise heterozygous, right? So how do we do that? When we cross with only the recessive parent, suppose in the next generation, if we get all these offsprings purple, that indicates that whatever here we have seen the parent dominant phenotype is pure homozygous. Suppose we do not get all four possibilities with the purple, we got 50% with one parent, 50% with the recessive. That indicates that the dominant phenotype is not pure. It is heterozygous, right? So let us check here. For example, you considered this as both dominant, okay? And we have considered the sperms from or the eggs from here. Clear? So now when we cross, we could get capital P, small p, capital P, small p, capital P, small p, capital P, small p, right? All are purple. But when it comes here, it is nothing but one capital P, small p, other capital P, small p, and two are small p's. That means 50% white, 50% purple. So when we get offsprings with 50% of one parent, 50% of other parent, it indicates that the dominant feature which it is expressing is not pure. It is heterozygous. So in this way, we can identify, plant breeders identify the unknown genotype. So what is the basic difference between backcross and test cross? Backcross, that means we are mating the F1 hybrid with any one of the parents. But it should be always with the dominant, then we call backcross only. Then test cross, we are mating the F1 hybrid with only the recessive parent. Even this is also back. That's why we do consider as all test cross or backcrosses, but a backcross is not test cross. The reason is here, in a monohybrid F1, when you cross the recessive parent, this is backcross and also a test cross. Why? Because we are going to test the genotype of that unknown parent and we are going to cross back with the parent. That's why we call it as both backcross and test cross. But when the hybrid is crossed with the dominant parent, that cannot be test cross because that is not used to test or to know the progeny with the parents, right? So that's why it is considered as only the backcross. Now, let us take few questions. Define and design a test cross. So what is a test cross? How do we define? It is a cross between F1 hybrid with recessive parent. Then, design a test cross. You can take one example from the garden pea, either with the tall and dwarf. Otherwise, we can take with the F1 hybrid of the purple and white color with a white color recessive feature. The test cross is between what kind of phenotypes and why is it used? Test cross is between F1 hybrid with pure homozygous recessive parent. Why is it used? To know the phenotype of the unknown species. Which of the following process will give tall and dwarf pea plant in same proportion? That means 50% one parent's tall and 50% the other parent's dwarf. No doubt, when we get 50-50, that means the dominant parent is not purely dominant. So we have to check where is that heterozygous. So check here, capital T, no way. We do get all tall. Capital T with small t, yes, definitely. Then capital T and capital T small t, no. Here also we do get tall. Then small t, all will be dwarf. So the answer will be B. To determine the genotype of all tall plant of F2 generation, Mendel crossed this plant with a dwarf plant. This cross represents. So when we have one hybrid crossed with a recessive feature, that is nothing but test cross, because backcross is with the dominant feature. Reciprocal cross is nothing but the character which you have taken in the first generation of the male, and in the second you will take with the female. Then it's not a dihybrid cross. Then some of the structured questions. Text crosses or backcross, but the backcross is not necessarily a test cross, right? So as we have discussed that test cross, here it's a backcross. The reason is in test cross, F1 hybrid is crossed with one of its parent, but that parent is recessive. That's why we call it as test cross. But as we cross back with the parent, we can consider it as backcross.